Hello, my name is Leland Pornschloegl, and my biology role model is Dr. Robert L. Evans III. So, who is Dr. Evans? Well, Dr. Evans completed his undergraduate from Juniata College. He then completed his graduate degree from the University of Minnesota Twin Cities. He is currently a postdoctoral fellow and works in the biophysical Wendy Gordon Lab at the University of Minnesota Twin Cities. Pictured on the right is Dr. Evans hard at work in the Wendy Gordon lab, and the picture is courtesy of Dr. Evans himself. So how did Dr. Evans get involved in science? Originally, Dr. Evans did not see himself as an academic. His parents were laborers and did not have expectations for his education. It wasn't until the 5th or 6th grade when he discovered science and it quickly became his favorite subject. His science classes, chemistry in particular, acted as a refuge for him in his other classes. He never had a concrete plan regarding his future career, but followed his friends to college, where he developed his work ethic, study habits, and graduated with a degree in biochemistry. The picture here of Juniata College is courtesy of the Juniata College website. What field of biology is Dr. Evans involved in? Well, Dr. Evans was previously involved in entomology and wished to study scorpions specifically. However, being unable to study scorpions, he is currently involved in studying biophysics, which is a branch of biochemistry. One of the projects that he is working on involves measuring the amount of force required to break the bond between two proteins, as shown in the picture here. This research can lead to potential medicines for diseases such as cancer. Both of the pictures on screen are courtesy of Google Images. So what has Dr. Evans been working on? Most recently, Dr. Evans has contributed to the article Discovery of Hydroxylase Activity for PQQB, published in the Journal of the American Chemical Society. As a structural biologist, he has been solving the structure of PQQB, an enzyme involved in the production of PQQ in many pathogens. The goal is to kill the pathogens or harm them by preventing the production of PQQ. The picture here is an image of PQQ and is courtesy of the Discovery of Hydroxylase Activity for PQQB paper. So what has he uncovered and what is next? Dr. Evans has achieved his end goal of solving the structure for the enzyme PQQB and has found the substrate's active site to show binding. More specifically, the substrate that he found is known as a substrate mimic, which is not the actual substrate used in the pathway, but mimics the actual substrate. His next steps would be to identify and capture the actual substrate before it reacts with the enzyme, which would be extremely difficult to do. In addition to being a structural biologist, Dr. Evans also enjoys bicycling, traveling, and writing computer programs such as Monte Carlo simulations in his free time. The image here is courtesy of Google Images. So what do I admire most about Dr. Evans? As an undergraduate researching and working with Dr. Evans in the Wendy Gordon lab, I admire the way in which he leads in the lab. I also admire the way in which he teaches the undergraduate students that he works with. He treats them as researchers and as his peers, which leads to a positive and more conductive work environment. Pictured here is Dr. Evans with the PI, Wendy Gordon, along with the rest of the staff of the Wendy Gordon lab as of 2018. This image is courtesy of the Wendy Gordon website. And here are the references that I used to create this video, and a special thanks to Dr. Evans for allowing me to interview him.